Hi children, our first unit is Life's Mysteries in Little Chamber. It is already completed in online class. Let us pass through the important points of this unit. This unit is started with the structure of a compound microscope. Let us observe the structure of a compound microscope. This is a compound microscope. The compound microscope have two types of lenses. Eyepiece and objective lens are the two types of lenses. This is eyepiece and this is objective lens. The lens in the condenser focus light on the material to be observed. This is the condenser, and the lens in this condenser focuses light on the material. Here the material is kept. The place where the material is kept is called a stage, and there is a clip in the stage. Stage and clip. This is condenser, and the condenser have a diaphragm which helps to regulate the intensity of light. This is mirror. Mirror is with a plane mirror on one side and a concave mirror on the other side. These are the structure parts of a compound microscope. Then when we want to observe a material with the help of a microscope, we have to prepare the observation material. For that there are different steps. First, take a cross section of a plantain root, then put the material in water to prevent dehydration. Then take thin sections of material and keep it in a watch glass containing water. Put the thinnest complete section to a watch glass containing stain. This is to get the color to the cell, and then add 1 or 2 drops of glycerin to the slide. Glycerin is added to the material is to prevent the drying up of the material. Then wash one of the stained section and place it in the glycerin on the slide. By using a needle, place a cover glass over it without forming air bubbles. By using tissue paper, wipe out the excess glycerin. Then place the slide and observe it through the microscope. These are the different steps for the preparation of the observation material. Then we are going to see the milestones in the history of cell biology. The first scientist is Robert Hooke, 1665. He observed cell with the help of a simple microscope. He called the tiny chambers as cells. 1831, Robert Brown discovered and named the center of cell nucleus. 1838, M.J. Schleiden found out that body of plant is made up of cell. 1839, Theodor Schwann found out that an animal body is made up of cell. 1858, Rudolf Virchow observed dividing cells and inferred that new cells arise only from existing cell. Then the important points of the cell theory formulated by M.J. Schleiden and Theodor Schwann are: the body of all organisms is made up of cell. Cells are the structural and functional unit of organisms. The functions performed by a cell are resist foreign particles, release energy from nutrients, and synthesize biomolecules. What is the difference between prokaryotes and eukaryotes? Prokaryotes are organisms without a nucleus in their cell. Example: bacteria, cyanobacteria, mycoplasma, etc. Then eukaryotes are organisms with a well-defined nucleus in their cell. Examples are amoeba, all animals and all plants. Let us start the second part. First, the part of the nucleus. Nucleus inside the cell. The nucleus has a nuclear membrane. Nuclear membrane is a double-layered membrane covering, and in nuclear membrane there are pores in the nuclear membrane. They are called nuclear pore, and the fluid part of the nucleus is called a nucleoplasm. There are spherical bodies that helps in the ribosome synthesis is inside the nucleus. It is called the nucleolus, and gene carrying network in the nucleoplasm are called chromatin reticulum. This is chromatin reticulum. This is the structure of a parts of a nucleus. Then let us observe the structure of a plant cell and animal cell. This is plant cell and this is animal cell. The main difference of plant cell and animal cell is, in plant cell a cell wall is present. This is a cell wall, and in animal cell no cell wall. Then vacuoles and plastids. Chloroplast is a plastid, and there are different types of plastids. The vacuoles and plastids are present in plant cell, and no vacuoles and plastids are seen in animal cell. Then in animal cell there is a structure called centrosome and lysosome. This is centrosome and this is lysosome. In plant cell there is no centrosome and no lysosome. These are the main difference between plant cell and animal cell. The other structure, that is endoplasmic reticulum, mitochondrion, Golgi body, these are common in plant cell and animal cell. Let us observe the different types of cell organelles in plant cell and animal cell. Mitochondrion, endoplasmic reticulum, ribosome, Golgi complex. These are common in animal cell and plant cell. Mitochondrion, it's a production and storage of energy. It's a powerhouse of the cell, which abundantly seen in the cells of liver, brain and muscle. Endoplasmic reticulum, it is a passage inside the cell for the conduction of the material. It provides firmness and shape to the cell. It is also called a cytoskeleton. Then ribosome, it helps in protein synthesis, seen attached to the endoplasmic reticulum or free in the cytoplasm. Golgi complex, it collects cell secretions like enzymes, hormones, mucus, etc. in small vesicle, seen plenty in glandular cell. Then vacuole, it stores water, salts, excretory materials, etc. Its membrane is called tonoplast. This vacuole and plastids are seen only in plant cell. Plastids are of different types: chromoplast, chloroplast and leucoplast. Chromoplast imparts color to flower and fruits. Chloroplast, we already know chloroplast perform photosynthesis and give green color to plant parts. And leucoplast, no specific color, seen abundant in storage cell. And centrosome and lysosome are the cell organelles that are seen only in animal cell. Centrosome, it contains centriole that helps in the cell division of cell, and lysosome, which contain digestive enzymes that destroy foreign substances entered into the cell. Then the examples for pigments, that we already seen that chromoplast is a plastid which imparts color to the flowers and fruits. The chromoplast contain pigments, different types of pigments for the impartition of color to the fruits and flowers. Xanthophyll, carotene, anthocyanin are the main pigments seen in the chromoplast. Xanthophyll, it is yellow in color, carotene yellowish orange, and anthocyanin red or purple color. Then the pigment that is seen in chloroplast is green. This is a flow chart of plant cell and animal cell. Plant cell contains cell wall, plasma membrane, protoplasm. Protoplasm can be divided into cytoplasm and nucleus. Cytoplasm contains cell organelles, mitochondrion, endoplasmic reticulum, ribosome, Golgi complex, vacuoles and plastids. And in animal cell there is no cell wall. It contains plasma membrane and protoplasm. Protoplasm can be divided into cytoplasm and nucleus. Cytoplasm contains cell organelles, that is mitochondrion, endoplasmic reticulum, ribosome, Golgi complex, centrosome and lysosome. Here vacuoles and plastids, here centrosome and lysosome. Other cell organelles are common in plant cell and animal cell. These are the main points of this unit. Complete the note and prepare for the test. Thank you.